Okay. So welcome. Hello. I'm going to, Eddie asked me, and you know this is the structure of this course, that occasionally other people than Eddie will give you a few lectures on the topic of your interest, or some particular topic, and so this will be 2 lectures by me, this today, and I think, if I'm not wrong, is it Tuesday? Thursday? Thursday. And then I think there is a jump because of this other meeting for next week. You know, next week we have this LSEC ICNC meeting. So the next one will be, I think, next Tuesday, 2 weeks from today, where 3 of you, I don't know who it is, will present 3 papers that I sent you. So I'll give you the background for these 3 papers, and we can discuss along the way, but then there will be the students who will present the topic. And so the general topic, and that's what I would like to highlight today and also on Thursday, is the issue that I think you came in a very good time for this particular issue that actually interests me for a long time, and I can say that maybe the title of this issue would be, how should I call it, maybe the impact of the single neuron on the overall dynamics of the network. So you know, if you come from physics, typically you assume that the individual element, let's say the individual entity, that's an electron or the molecule, has a very, very small effect on the overall behavior of the network. So you can really represent the element in a very simple way, and the emergent property of whatever system you study is a result of the interaction between many elements. And so the classical question that is really, really classical, and there is a tension between the individualists like myself, who believe that the single neuron has a very important impact on the society, and the physicists, the classical physicists will say that society, when I mean society of neurons, or the behavior of the society is really an emergent properties of many interactions, but the individual property of the individual cell or the individual synapse is really not very important. So I can simplify the entity, so to speak, but I can look at the interaction. So let's call it the fight between individuality and society, something like that. And so so this was an open question for many years, and it's still partially an open question, but I think in the last few years, really last year and the year before, this year, there are some critical experiments, really beautiful experiments that I hope we should get to. Some of them will be represented by the papers that you represent, and some I will try to highlight here maybe. But in order to do this, I have to give you some foundation of the background. But this is the context. So you will know where I'm trying to head, or what is the issue I want to discuss. And I know that some of you studied it, so what I'm going to speak about, so I want to very fast go through cable theory and compartmental modeling, just in order to give you my intuitions, which is, I think, a good speaker should do, the intuitions, not the mathematics now, but the intuitions. And this takes time to develop, so my role will be to transmit intuitions to you rather than the equations, because the equations you can use elsewhere, but intuition, it takes time to develop. So if I succeed to highlight some of my intuitions, which you also should suspect, of course, because my intuition is not my intuition, but it should be useful. So my role in the next 2 hours is to provide you with intuitions about how do I see single neurons as a computing device. Okay, so I'll give you a little introduction about neurons and about the fight between Cajal and Golgi. You should know, as scholars in neuroscience, you cannot avoid this big fight 100 years or so ago, a little bit more. And then I want to highlight some new intimate acquaintance with neurons that emerged through these new technologies, several new technologies. And again, if you know a lot through introduction and so forth, it will be easier for me to run faster. So you please give me feedback about what is unnecessary. Okay? really feel free to, because I don't really know each one of you exactly what you came from and what you know. So I want to highlight the passive cable theory of roles, and then ask what kind of questions would you like to ask using this cable theory. It could be passive or not, or nonlinear later on, and some insights about passive cables, about nonlinearities in neurons, and eventually some project that we are involved in. So let's see. But stop me again at any point, ask me questions, and so it should be very free exchange of intuitions. So everything starts in 1906, actually before, but this is the classical year because this is 5 years after the beginning of Nobel Prize. So the first Nobel Prize in 1901. This is the first Nobel Prize in neuroscience. So Ramon y Cajal and Golgi were two giant anatomists. Probably no one ever approached this level of being anatomists. They have schools, especially Cajal have already grandchildren in Spain and other places that are continuing his love for anatomy. I could say love for anatomy. He was a painter, he was a womanizer, he was a fighter, bullfight. He was a very interesting character. Ramon y Cajal was a real character, and Golgi was less of a character, but also a genius in the sense of developing technologies like the Golgi stain. So we still use today the Golgi stain, which I'm not going to go into the details, but it's a stain that enables you to pour the stain onto the tissue. Some of the cell will pick up the stain, some not, so you will get a disperse, you will get a random coloring, and this is the technique that Cajal was using to see cells in a very fine details. And Cajal, as I said, was also a very good painter. He wanted to be a painter himself, so he drew the cells, beautiful cells, and I'm sure you know them, classical paintings of the Purkinje cells and other cells. So he has many volumes of beautiful paintings of. And if you're interested in the beauty of neuroscience, you should really read the recent book by Javier de Felipe, who is a grandson. When I say grandson, I don't really mean grandson. I mean intellectual grandson of Ramon y Cajal, who is continuing this work, but of course with new technologies. So there was a huge fight between these two. On the podium of the Nobel Prize, they wanted to kill each other physically, because Ramon y Cajal said that neurons are individual entities, separate from each other, and Golgi said, no. He was a romantic, he liked the operas, and he said, the soul and all these kind of things that cannot be separated. We are one unit. The brain, unlike other cells in the body, are connected, physically connected. They didn't know, of course, about synapses. So there was big fight between the two, where the cells are cells also in the brain, and the term neuron was a new term that came following the success, of course, of Ramon y Cajal to win over Golgi by convincing us all that there are neurons, that the brain is built from separate entities called neurons. Sorry? Yes? Speak.
It's like the mouse conflict regarding whether there is in the brain a functions separation.
Say it again?
Is there in the brain modularity?
Modularity. You ask whether there is separate modules in the brain, which is another level, of course, of discussion.
Yeah.
Could say. There is some similarity to that, but it's more kind of a refined similarity. Because here we talk about who is the elementary entity. So Golgi said that there is this what he called syncytium. He spoke about syncytium, which is a word we don't use today anymore. He spoke about physically connected network of neurons that are physically connected, and they together subserve an emotional function, for example, the love for Verdi. So he was speaking like this, in this term. And Ramon y Cajal said there is no syncytium, each cell is separate. By the way, he could not see it with his microscope. You know that the microscope that they were using 100 years ago was extremely poor. Even today, of course, you cannot see with light microscope a synapse. I don't know if these two cells are connected or not, but with this technology even today. You need an electron microscope, which is certainly not in this period of time, but he failed from different reasons, and I don't want to go into his arguments, because he could not really make the argument kahal, that he sees that they are not connected physically. He cannot see that. But he had some indirect evidence. He killed one cell and he saw that others are not being killed, so maybe I'm individual and I'm not affecting the other, and I stain one and it doesn't jump to the other. So I don't want to go into it now because this is really classical history, but I wanted to start the lecture by history. Because it really all started here with the question whether we have in the brain, like in other areas, like in the skin, people knew that there are cells and they are separate. But here they did not know that there are cells and they are separate, and he claimed this, and this was a difficult claim. For us it's very natural, but it was a drama, the neuron doctrine. So the neuron doctrine won over 100 years ago.
But it's not, maybe it's not just Monica Hall was right, but also Golgi was right, because Monica Hall only wanted to argue that they're separate, but didn't emphasize the connectivity.
Exactly. So today we know that there are networks. I agree with you completely. We know today that ensembles of neurons represent things and not the individual one and so on. But the issue was about physical. Is it a physically connected network? And the question of physical, you can say that the synapse is also a physical thing, and chemical connection is also chemical. But he spoke about this in situ, and he had a very particular sense then. It was really physical, electrical connection, maybe I could say that. And electrically connected. Continuation. It's a continuation of one big thing. And
also
electrical connection.
Yeah,
but not in the cortex. So, but yeah, you're right. It's not very clear what was the fight about then, today. But then it was a big, big fight. Is it built from entities that are separate? Each one is doing his own thing. For example, signals are propagated first in one and then transmitted to the other, or there is always something together. You can look at this very beautiful book by Gordon Sheffer, The Neuron Doctrine, where you can see the whole process of developing the notion of the neuron. The term neuron was not, by the way, invented by Ramon Cajal, but by a German physician a few years later. But following the work of Ramon Cajal, there was this notion of neuron. The term neuron, nerve cell neuron, was not before Cajal, because they did not think that there are cells in the brain, separate cells in the brain. So again, you're right, there is this issue, what exactly each one meant. I don't want to go into it now, but I just wanted to highlight that there was this big fight. We should know about this big fight and the big winning of Ramon Cajal over Golgi then. Today we are not anymore so sure who is the winner, if we want to speak in terms of fight. But anyway, so this was interesting at that point in time. You know that Freud was involved. Freud himself, not Lucian Freud, but Freud, the grandfather, was thinking that cells are separate, and from this he developed his theory of personality, because he left neuroscience. He was a neuroscientist, Freud. He left Trieste. He went to become psychoanalysis. He developed theory of psychoanalysis. It's all connected to this. But so this is a very nice book, the theory of the neuron doctrine. So, but again, I'm not going to go into it, just to tell you that this was a very active times with Freud and other German physicists and physicians, and Cajal and Golgi and others fighting about whether there are neurons or not neurons, individual or not individual, network or not network, but very active time.
How single neurons are related to psycho?
So intuitively you say, if I'm a single neuron, yes, in this audience, I'm a single neuron. Yes, Freud, by the way, Freud was also a very good painter, and there are some beautiful paintings by Freud about cells. This is in the case of the lobster, I think. And so he said, no, each cell is individual, that's his claim. Each cell is individual, and each cell has a function in this particular animal that he studied in Trieste. And because individuality, I'm going to go back into it, yes, but in a modern term. If individuality is so important in generating the behavior, I will switch into the individual person and discuss how you, I'm not Freud, I'm saying what Freud was saying, yes, was motivated by. that individuality is very important for the generation of a society, but the nature of the individual is important. Anyway, so we don't want to stay in the history. I want to go into the modern time, but I just wanted to give you a good story as a beginning, yes?
So, conceivably, suppose Golgi was right entirely, so there was just one big centralized entity. Could it not have done the exact same thing?
No, because, for example, we know today that synapse, the chemical synapse, the fact that you have a chemical connection, not electrical. There are electrical synapses as well, but most of the synapses are chemical, and most of the, for example, plasticity effects, as you know well, and most of the drugs that we are using against, I don't know what, depression, are working on the connection between separate cells. So if it would have been a syncytium, and you will pour a particular drug, you cannot expect a separate effect on separate entities because they are not connected electrically, but they are connected by something, apparently not by chance. Because you want to modulate the system, and the way the system is being modulated, our system, very powerfully is by the fact that the connection between cells, which does not exist in the syncytium. The syncytium is a physical connectivity, which apparently, I guess you can design a system that is also flexible with electrical connectivity, but I think that what Cajal wanted to say, that because you're individual and you are somehow connected, he knew that they are discussing, he knew that they are communicating between each other, he knew that they are not just sitting connected, but he didn't know about synapses. But he said they are not physically connected, so somehow they interact, but he doesn't know how. This came later, many years later, the notion of synapse. And this is 20 years later. So it's not the same system. A system that is separated with an element, with a communication element that you can control locally, is different than a system that is integrated electrically, and it's more difficult to, so to speak, manipulate each one separately because there is no each one. It's one thing. But we can discuss it later, but really give me time to continue because it will be an historical lecture, which would not be a bad idea, by the way, to give you the history of neuroscience. But there are several good books about that. Not only the history of the neuron, the history of the neuroscience thinking about regions, about the specificity and so on. But I'm not going to do that. And I'm not going to talk about Van Gogh, who at the same time that Ramon y Cajal drew trees, Ramon y Cajal drew beautiful trees, Van Gogh also drew beautiful trees in Arles. So you can go to Madrid, for example, today, and you can see what you cannot see very well with this slide, but these are cells that were drawn, that were stained, not drawn, sorry, stained. Were stained by Cajal, and Cajal was really seeing this in his microscope, and he was painting, you know, the axon and the pyramidal cell dendrite apical tree. And you can see there is a Cajal museum. That you can go and see. Really was beautiful. But you really should buy, if you like aesthetics, you can buy the book of Javier de Felipe. Okay, so these are the drawings of Ramon y Cajal. We saw recently the retina. If you heard Michal Rivlin talk, but this is all fantastic drawings, really only by microscope and by hand. Very, very beautiful. And one aspect that I should maybe mention, just for you to know, that there is one classical drawing by Cajal with these arrows, and this is another imagination of Cajal, because it's only imagination, all this is imagination. He thought that signal propagates, whatever signal, some signal propagates from the dendritic tree, through the cell body, through the axon, to the other cell. So he spoke about propagation in the network of separate cells. So these arrows wanted to show that there is this that the neuron is a one-directional flow of signals from some input source. So this is from the thalamus, for example, an axon from the thalamus that he reconstructed and drew, connected somehow, because there is this arrow from the axon to the dendrite and from the dendrite to the cell body. So there is some direction of flow. So that's what he called theory of of dynamic polarization. The neuron doctrine is the theory of dynamic polarization. So these are the two theories of, if you can call it theory, theories of harmonic Halley, the neuron doctrine theory, and the dynamical polarization, which means the dendrites are polarized first, and this polarization propagates this direction. Okay, so this is very classical, and now today, of course, you know that we, for the first time after many, many years, about 10 years ago, or a little bit less, now we can systematically record from dendrites, something we could not do before, because all these new optics, you know, big video microscopy, 2-photon microscope, and all this new staining, so you can really visually directly patching the dendrites if you want, maybe even at several locations, and you really start to see if there are connections, where does the signal flow to and from, and all these kind of new technologies. And you should hear Michael Heuser coming next week, Thursday next week, which is a very interesting day because it will be Larry Abbott and Michael Heuser and Sackman and others talking, and he will speak about in-vivo computation, Michael Heuser. And you can reconstruct cells, and just to highlight the things that you may have seen, because we are usually like to think about them. So if you had a polarized glasses, you would see the three-dimensional cells here as they look in the brain. So the dendrites are the green tree, and the axons are the red tree with the yellow spots. And so you can see, of course, that axon and dendrites are huge, bushy trees, and these are only local axons. Of course, there are axons going elsewhere, but this axon have a lot of these yellow spots, which are the boutons, which are the site of presynaptic vesicle release site. So you can see that each one of these little spots, these is boutons, are the presynaptic site that connects to the spine in this case, because this is an excitatory cell, to the spine of the postsynaptic cell. So just to give you an impression of the complexity of this element that is called neuron. Okay, so about 5,000 or so boutons, 5,000 contacts being made from an axon to its friends.
The reds are axons?
Yeah, the red is an axon. The process is the axon itself, and occasionally, about on average every 4 microns, you will see a release site. A synapse,
so
a presynapse. Yeah.
So it means that there are other neurons.
Yeah, of course. So there is a bush, there is a jungle of neurons, and one of the dendrites from one of the neurons will go here, and will receive an input from this particular release site, or putative release site. Yeah, it should be functional. Just to give you a view of the element that we deal with. And so I said today, and there are some world experts. We don't have one like this in Israel. There are world experts in the world. All of them are students of Bert Sackman, who will be here next week, that developed this amazing technique that they can record simultaneously from the same neuron at different locations, patch dendrites and patch the soma, and they also now can patch usually one spot in the axon, and start to better understand the relationship between what happens in the dendrite to what happens in the soma. Simultaneously, because they have all these electrodes. Classically, of course, in the last 30 or 40 years, we could only record from the cell body. So we could only see the activity that is summed up from all the dendrites here, and the spikes that emerge here are reflected in the cell body. But it's just one point of view, and today we can have several points of view using this technique, and we also have the 2-photon microscope, which is really shocking technique. I'll show you in a second a paper that will be surveyed by one of you, which is really showing the input itself per synapse, or the orientation selectivity of the input itself to a neuron, which is a really huge jump. So this is the jump by Connors, who will give us Heller lecture in January. I think this is the most interesting paper by far in my field, at least. I think it's in most neuroscience. The most interesting paper, because the technology enabled for the first time to do what we were expecting for many years, not only to look at the output of the cell, meaning recording from the cell body and seeing tac, 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 tac spikes, but also recording the input to the cell, meaning looking at the synaptic input. This is an amazing technology, and this is what emerged from it, but I'm not going to talk much about it because this is one of the papers that I gave you by Connors to read. But Connors succeeded to develop, you don't see it very well, but he used the two-photon microscope and calcium imaging, which Eli Nelfin and Adina Rabi are using, which is now a very classical way, but most of the imaging of calcium imaging activity is done from the cell body. So you see when the cell body fires a spike, or the axon fires a spike actually, and the cell body repeats this spike, you see calcium going in, and so there is calcium in the cell body, you see the cell fires. But you don't know what happens here locally when the input is activated, and this Connors developed a very fine two-photon microscope resolution that enables you to look at calcium imaging locally on a dendrite. Why in vivo, while the animal is seeing something. And so this is really a renaissance time for dendritic people like me, because for the first time we can really see the input to a particular cell when the cell, so to speak, the animal, let's say in the visual cortex in V1, looks at moving gratings in particular direction. So you know that the cell, let's say the cell classically is, let's say, 45 degrees orientation selective cell. This is on the output. But what is the input to this cell? And this is what is shown here. That you can record the orientation selective of the input of the synapse, of this synapse. This is the orientation selective of this synapse. And this is the orientation selective of, I don't know, this synapse and so on. So you can look with two-photon microscope, and this is only done in Germany today because nobody succeeded yet to refine the two microscopes to the level that you can see the input in addition to recording from the output of this cell. So let's say the cell is 45 degrees orientation selective, but you can also now map not all the synapses, but many of the synapses in terms of the input to the cell. And you try now to understand how this barrage of inputs eventually is computed, so to speak, in order to give a particular sensitivity to this orientation. Is it stable? Can you plastically change it and all this kind of thing? But you can really look both at the input and the output. This is a real jump in neuroscience, a real jump. See, at last you see what is the network telling me. Okay, so I'm not going to go into it, but I'm going to just highlight some of what I feel is important for you to remember. But again, if you know all this, give me a hint and we'll go fast because there is a lot of material and I only have two hours twice to talk. It's certainly not enough. So, just to give you numbers or general kind of a scale of numbers or dimensions, when we speak about morphology of neurons, you have to remember that neurons, so the diameter, if you speak about diameter of cell body, for example, or of dendrite, it will be in a micron level, yes? So the cell body could be 20 microns, 10 microns, the diameter. The dendrites near the soma can start, for example, the apical dendrite of pyramidal cell could be 4 microns in the beginning, then it becomes thinner and thinner and thinner and thinner until it goes about 1 millimeter away, so about 1 millimeter away, and then you will see dendrites that are less than a micron, let's say 0.3. And that's it. You don't get much thinner dendrites. So this is the scale of diameters.
You don't see them.
No, but you can see them in electron microscope. And you see that they end, when they go too thin, they finish. They don't go to become too thin. Of course, the thinnest thing in dendrites are the spines, which are part of the dendrite, the dendritic spines. And so you know how dendritic spines look like, yes or no? Yes, so that you have a dendrite. Some dendrites, not all dendrites, are spiny, which means that they are covered with these unique structures. And this is usually, mostly, excitatory neurons have spines. Inhibitory neurons are typically smooth. It's not the case for Purkinje cell. For example, Purkinje cell is an inhibitory neuron, but it is covered with spine. But in the cortex,
You're talking about spines in the dendrite.
dendritic spine. Yeah.
So this neuron is not only excitatory or inhibitory, it can get inputs which are excitatory or inhibitory.
No, it can get input, but the output of this cell,
Yes.
that a cell is excitatory, yes, we have to agree, that when I'm saying a cell is excitatory, I'm allowed to say that the cell is excitatory because all its output, not the input, all the output.
Saying that this affects the spines, which are the input structure.
Yes, but I'm saying if I'm a cell, what do I say to my environment? I'm saying excitation to all.
Okay.
I'm saying excitation. I release glutamate. So everybody interprets me. All my listeners interpret me as excitation.
Yes, but then you said that if you are an excitatory cell, then your spines have
No.
structure.
No. So maybe I missaid it. I said that excitatory cells, by my definition, typically have spines.
I understand, but it's strange that your output affects the input structure. Because
This I didn't say that the output affects my input structure. I said that there is a correlation between the anatomy of the dendrite, which is a spiny anatomy.
Yeah.
So all pyramidal cells in the cortex are excitatory, and all of them have spine. All spiny cells in layer 4 are spiny cells, and they are excitatory. All the rest of the cells are smooth and they are inhibitory. So there is a stronger, in the cortex, there is absolutely one-to-one correlation in the cortex. Not speaking about cerebellum. In the cortex and the hippocampus, if I look and I see spines, I know, by some miracle, that it's also excitatory. So that's an important thing. Why it is, that's another issue which I cannot explain, but it affects. So another thing is that the spines are getting excitatory synapses. So most of the excitatory synapses on spiny neurons are going onto the spines. It doesn't mean that there is no excitatory synapse here, sometimes, and most of the inhibitory synapses, so this is inhibition, do not go to the spines. But there are some spines that receive both excitatory and inhibitory, about 10%. So the architecture of connectivity, if you are an excitatory cell, is first that you have anatomically spine. Second, the spines typically receive, there is no bare spine. It doesn't sit there. If there is a spine, there is a synapse. And if there is a synapse, there is an asymmetrical synapse, which is excitatory. So this is the typical case, and this is not the typical case, but it also exists. And if you look at the dendrite, typically you see inhibition, but you also see excitation. So I'm just telling you now a morphological fact. Okay, it's a morphological fact. What is the meaning of this morphological functional meaning? That's another thing. I'm just telling you who is the character you deal with. The face of neurons, yes? The characters of neurons, the anatomical character of neurons, and also the physiological character, because I spoke about excitation and inhibition. So again, you will get all this, so you don't need to write everything, but I'm just giving you some of my, I can call it intuition, or the way I think about these characters that are called neurons. Question? Okay, I also want to highlight that there is electrical aspect, of course, to neurons, and typically when you speak about electrical characters of neurons, you speak about the passive part of the neuron, character of neurons, which means the membrane properties, the resistance per unit area, which is Rm, these are the units, and the Ri, which is the axial resistance along the dendrites. There is an axial resistance, Ri, which is this number, and there is the membrane capacitance per unit area, which is Cm. These are numbers. So, for example, if I multiply Rm by Rm by Cm, I should expect the time constant of the membrane time constant. Okay, so these are numbers, and you know the membrane time constant of neuron can range between 1 millisecond to 100 milliseconds, depending on the type of neuron. So these are the passive properties on isolation, yes, so to speak, without any activity or any nonlinearity. And you can speak about the space constant, how long electrically are dendrites. This is a range of what is called lambda, the space constant, and all these other things that I don't know now to go into it. So these are the passive properties. The input resistance is another passive membrane property. Just to remember that if I impale the cell body and I inject current, steady current I will get some voltage V. And so the ratio between them will be the input resistance, which may go to 100 mega ohm, maybe even giga ohm, depending how small the cell is and so on. So these are numbers here and so on. I don't know if we go all over it, but for example, if I would impale with an electrode on the spine head, if I could do it, and people are starting to do it, if I could impale with an electrode here, which is very, very difficult because this is very, very small. But people start to patch dendrites, patch spines. Dendrites I already showed you, but spines are even thinner. By the way, I mentioned dimension, and when I said that the thinnest dimension of neurons is the spine, I meant the neck of the spine here, which is a little branch. This little neck is about tenth of a micron. So that's the thinnest process that you can see is the neck of spine, but it's part of the dendrite, so it's a branch. It's a little branch with a particular, and this could be 1 micron in diameter or something like that. So anyway, so all this is written here. Just want, of course, to mention that there are excitable channels, nonlinear channels embedded in the membrane of dendrites. So dendrites are certainly not passive, but nonlinear, active. And there are all this huge amount of channels. There is now a very beautiful project by Henry Markham. It's called Channelpedia. The Channelpedia project, very important project, because we don't really know to characterize these channels. I mean, when I say characterize, I mean the dynamics of the channels, the voltage dependence, the time dependence, voltage dependence, like the Hodgkin did for the sodium and potassium, voltage and time dependence. There is this Channelpedia attempt, which is done by robots, to really take a gene, the sequence, a channel, a protein, put this gene into a bare cell, grow the channels there, and then patch, so to speak, by the robot, and characterize the dynamic of this channel. So we know that there are about 500 different type of ion channels in the brain, about 500 different type of ion channels. They have names KV3.11 and so on. These are all these gene names, but we never characterize the excitability or the voltage dependence of these channels, and this is a project being done now, because you understand that this soup of channels, that is a soup of channels, many combination of this soup of channels in the dendrite generates the nonlinearity in the dendrites, and this nonlinearity, I try to do, it has a strong effect on the working of the cell and thus of the network dynamics. And it's interesting that the dendrites are so rich, some region, after all the channels are being generated at the cell body and being sent from the cell body to the axon, because the axon has to carry spikes and do other things, release and so on. And the dendrites also have to do things, and apparently most of the excitable channels are in the dendrites. And the different dendrites have different combination of this soup of channels.
Most of the types of channels?
Most of the type of channels of these 300 or 500, I'm very clear, are in the dendrites. So somehow the cell body cares, so to speak, to send this part of this type of channel there, pulse tune dependent, low threshold, and this H current here, K current, A current there. We don't really understand why the nonlinearity is so rich in dendrites. There are no big spikes, but there are some spikes in dendrites, but there is a lot of nonlinearity. So this is the physiology of dendrites, is not well understood, because it's very hard to really, so to speak, map the local nonlinearity in each branch, but it's a very rich nonlinear system probably for some good reason. But what is this good reason? It's not clear. There are many theories. Maybe we'll go into it. So this is this excitable property that is very shrinked here, but this is a very, very big effort today to characterize this Channelpedia. And of course there is synaptic mapping, synaptology, you can call it, the synaptology of dendrites. And as I mentioned, there are, of course, and you know well, there are different types of synapses. Of course, the most pronounced distinction is between excitation and inhibition, which is also not so trivial to think about this dichotomy. But apparently there are these two types, and these two types also have an atomical signature, which is another amazing thing. Why can I say symmetrical synapses are almost always inhibitory, and asymmetrical, which means that by EM I see some asymmetry. So when I look at the anatomy of synapses by EM, by electron microscope, I see some structure, and this some structure I can call asymmetry, and this asymmetrical structure is always almost associated with excitatory synapse. So just by looking at the synapse, anatomy say, this is excitatory synapse because it's asymmetrical. This is inhibitory synapse because it is symmetrical in electron microscope. So there are these two general type of synapses, but of course there are this other type of receptors which interpret the input. For example, excitatory input could have NMDA or non-NMDA receptor, and this creates a certain time of dynamics for the synapse, and a certain conductance change due to the synaptic activity and so on. So there are these different receptors who take, so to speak, who receive the transmitter and interpret the transmitter to be excitatory and inhibitory, because it's not written on the transmitter, of course, that I'm excitatory. Sorry, nobody tells me that glutamate is excitatory. I'm just saying that in the brain, glutamate is interpreted because of the reversal potential and so on to be excitatory, but there are different dynamics to this particular glutamate because of the receptors that are different, and also the GABA A and GABA B receptors that have different dynamics and kinetics for the inhibitory synapses, but this is a very rough kind of distinction. And of course, there are many subtypes that we know today about the type of synapses, and there is a very interesting fellow who is studying the development of synapse with evolution, and you can see that our synapses, for example, the NMDA synapse, which also exists in mouse and also in lower mammals, the NMDA synapse is much more rich in terms of protein involved in our Homo sapiens brain than in the brain of, let's say, the monkey. So the synapse also has developed from a very, very primitive synapse, let's call it, to a very sophisticated synapse in terms of the proteins involved when it is activated and the many states that it can go to. I'm not an expert in this, but it's another issue of interest, the development of the synaptic input to enrich plastic properties and other properties of the network.
So, when you said excitatory synapses are asymmetric, is that just the anatomy? You look at it and it looks
Yes. I didn't bring an EM microscope, but if you would look at an EM microscope in a high magnification on the synapse, so you will look, let's say, you will see the axon. So this is your axon, let's say, with this bouton that I mentioned before. There will be vesicles here, yes? And there will be, let's say, the spine here, or not the spine, depending where the synapse is sitting, something like that. And you will see in this case, when I say asymmetrical, when you zoom in with an electron microscope, high resolution, you will see, for some reason that we don't understand, why is it connected to the function, you will see in the microscope, you will see highly dense material here in the receptor side, and less so in the presynaptic side, so you will call this asymmetrical synapse. Just by looking at it, you will say it's more dense on this side than on this side, and if you look at another synapse, which apparently is an inhibitory one, you will see that both sides look symmetrical in the density of electron microscope. So anatomists, they just look and they say, oh, this is symmetrical, and this is not symmetrical, and this is symmetrical. But now we know that there is a connection between the structure of the synapse and the function of the synapse in terms of is it inhibitory or excitatory. Why? I don't know. They also speak about difference a little bit in the shape of the vesicles, you know, between inhibitor and excitator. So, but again, this is like a miracle of some sort, because it could be different. Why should nature decide that vesicles and this shape is inhibitor? Something about the transmitter that has to be embedded here, so there will be a GABA transmitter here, there will be a glutamate receptor transmitter here. Maybe this is part of the asymmetry, who knows what. I certainly do not, but we should speak with anatomists about their explanation for the asymmetry or symmetry of synapses.
So it is denser at the postsynaptic side? Or?
I think in the postsynaptic side, but maybe I'm wrong here. I have to check again. I have to check again where the asymmetry, I think it's in the postsynaptic side, but I'm not 100% sure. I'll check for you. Okay. So another controversy came, theoretical controversy, came from these two people around the 50s of the 20th century. So John Eccles is a Nobel laureate, worked a lot on the spinal cord. Wilfrid Roll is a physicist, theoretician, worked on the Manhattan Project, came to work with Eccles and saw dendrites. Everybody saw dendrites because dendrites were known since Ramon y Cajal. But Eccles, in all his work about the spinal cord, ignored mathematically, so to speak, dendrites and considered cells to be isopotential, point neurons. He said, okay, there are all these dendrites. We know that there are all these dendrites. But as a first approximation, let's ignore them and analyze the signals that I record from the cell body, assuming that my model is of a isopotential cell. Rawls said, no, you cannot ignore it. Now, why is it so important? It looks again like, why do we need controversy? There are enough controversies. So everybody knew Ramon y Cajal, Purkinje cell that I mentioned before drawing. We know that the dendritic tree is a huge bush and that the synapse sit here. But when you do that, in all the drawings from McCulloch and Pitts and every other most books drawing, you can see that the dendrites are shrinked. They disappear because it's hard to paint them, but not only because of that, because you believe that as a first approximation you can put all your synapses directly on the cell body, functionally. So you can shift all your synapses, so to speak, lump all your synapses into the cell body, and you can analyze, for example, the electrical signals you record from here, assuming that it's a point neuron. Now, what does this assumption mean? And that exactly was the mistake of, really big mistake of the Nobel laureate Eccles, because when you assume that this is an isopotential cell, there is no space, it's an isopotential cell, it means that voltage, this in the passive case, voltage will develop exponentially if you inject a square current pulse. Because it's an RC circuit, yes? I hope that I don't need to go into this. Yeah, it's an RC circuit, and this RC passive circuit will respond to a step current by exponential growth and exponential decay of voltage. So you can recover the time constant of the system if you think it's an exponential decay. Through this assumption, you can recover by plotting, the ln V against ln T. So the slope should be your time constant. But if it's not isopotential, meaning that most of the current that you record from here, when you inject, for example, through your electrode, or when you see an EPSP coming from the dendrite, most of the current that you record from this membrane is escaping elsewhere. So most of the conductance is not local, it's a distributed system, so if I inject a square pulse here, most of the current goes elsewhere, because it's not isopotential. Then it's not going to decay anymore exponentially, not growing and not decaying exponentially, but it's a multi-exponent, the infinite sum of exponents, and there is a huge underestimation of the real time constant. The time constant of cells are very important parameters to understand the temporal summation of neurons, and so all the calculation of Eccles were completely underestimation of the real time constants of neurons because of this model assumption that you can assume that it's isopotential.
Underestimation means it's actually
larger. So the real time constant is larger than what you assume. Okay, so if you have an isopotential cell, your voltage, and you inject, let's say, a square pulse here, you will get this exponential and decay and growth. It's 1 over 1 minus exponent, and this exponent me to the power of t divided by tau. But this is for this kind of cell. But if you have a cell that has a structure like this, for example, a cable theory of all, then if you do that, you will get, because of these other time constants, the equilibrium time constant that really equalize the voltage from here to here, which are faster than the time constant, you will get something like that. And only at the tail, if you wait enough and your cell becomes isopotential slowly, after you inject the current, the voltage is high here and low here, and then slowly they become isopotential in infinity, then the slope here is the real time constant of the membrane. Because at this time, the current can only flow through the membrane. But in the beginning, most of the current flow axially, which is faster. So, for example, Eccles took this to be an exponential, and so it got a much faster time constant than the real time constant, just because he assumed that it's isopotential. a model mistake, a brain model mistake. You assume something that is wrong and you analyze your data. So this is one problem. There were other problems. And one of the major problems, so this is the, anyway, so there was a very interesting, so you should see how egoistic scientists, and also they think they know everything, scientists are very sure of themselves. So Roll was working with Eccles. Eccles was a Nobel laureate. It's not simple, yes? And Roll drew this just to show to Eccles that just because very simple electrical, considerations, just because of this, of the area of the soma and computation of this axial resistance, 95% of the current you will inject goes there. Just because of the structure. We didn't do anything bad. Yes? But the implication was what I just said, that you are wrong in estimating the time constant of this system. So this is a drawing by Roll, trying to show that current flows away from where you inject it. Eccles decided, and then he wrote the cable theory, which is a beautiful paper from '59, and Eccles decided not to allow this paper to appear anywhere. So because he was a Nobel Prize, he wrote to all the journals, this is a bad paper, a wrong paper. This was disputed, yes? It's not easy to recover after that, but eventually there was a new journal that Eccles did not know about, and Experimental Neurology, 59, the first volume. So they accepted the paper, became the most important paper in cable theory, and he came out from Eccles, but they never spoke since then, and so on. You can see how scientists can be mean, because it destroyed a little bit of his work before. The analysis was wrong. Anyway, so you should be aware of scientists. Very aware of scientists, again. Really very hard to distinguish between their work and mistakes and themselves. If I'm mistaken, it means that I was nothing, which is the wrong way to look at it, of course. So here is Roll. And so Roll was a physicist, of course. As I said, and he wanted, of course, to simplify his neuron model, because he didn't really want to compute all these little details about all these things, and he suggested a systematic way to reduce the complexity, mathematically. To take a complex neuron and mathematically reduce the cell, and the question is, how much should I reduce it? And of course, the answer is, what question do you ask? Yes. But maybe it's not so important that you make a mistake in the time constant, or maybe you compensate for the erroneous time constant if you put it here. There are many other issues to decide what level of model am I taking to represent my network or my system. Of course, it depends on the question. But anyway, there is a systematic way to reduce it. And so, in my way of thinking about this, I think about this type. You know, by the way, that Cajal called the pyramidal cell the psychic cell. So this is the name of Cajal to this cell, because he knew that it's in the cortex, and the cortex generates psychic, and so he called it the psychic cell, the pyramidal cell in the cortex. And so I think this is, so people today, myself too sometimes, when needed for a particular question, I model mathematically using compartmental modeling all the details. So then I look at myself as being Rodin, yes, Rodin, because Rodin modeled the kiss in very fine details, but still it's a model of a kiss. So this is a model. I can model this using compartmental modeling approach, also cable approach under some assumption. And if I become a reductionist, so to speak, and I reduce my complexity, I think eventually I like to think about myself or everybody else as being Brancusi, because also Brancusi modeled the kiss, you know. This is a reduced kiss. Still a kiss, yes, because you keep the kissiness of the model, but it's the minimal model, because if you a little bit reduce it, you lose the kiss, so you lose the function, yes. So we are somewhere here always when we model cells. We have to decide what level of details of modeling or simulation you want to go, and the question is very deep, because the question is how much this reduction, what do you lose while you do this reduction? This is of course true in any field of physics or mathematics when you do reduction, but you have to be careful that you know what you lose. But in order to know what you lose, you have to know what you have in the full one. So you have to have the full kiss and the pleasure of the whole kiss, and then you get a reduced kiss. So that's how I think about modeling neurons in general. That I have to decide where am I, so to speak, between Rodin and Francusi in terms of what am I asking, what kind of mathematical or theoretical questions do I ask, then I choose the correct level of description for this particular question. And so if I had time, I would argue with you about the Blue Brain project that I'm very much involved in. The Blue Brain project is really at this level. It's a really brute force of taking every detail you can think about, put it together in a large, heavy supercomputer database and also simulation. This really annoys many people in the world, as you know. To me, it is not annoying at all. I think the world is going to detailed simulations, not only in brain, but in every aspect of life, including the single cell and atmosphere and physics. Very dense, high simulations in details. It's called the fourth generation. There's a very nice book about the fourth generation of science. So there was theory, modeling, simulation, now database simulation, fourth generation. I'm not going to go into it, but this is the big controversy in science today, as you know. Am I going to use all my computer pilot to run simulation without asking questions, or should I come with an hypothesis and then run simulation or modeling? I'm not going into it. This can take the whole, we already did it once with Chaim and Gedi, maybe we should do it again. I'm not going to go into it, but I'm saying as part of this, that beyond the single cell simulation, with details or less details, there is the issue of network simulations, with details or not details. For example, the whole cortex, the whole brain, the whole I don't know what, the whole in details. And so where are we today? Who is against who? It's an interesting, I think, historical moment where we have the power to simulate life, to simulate life in a very fine detail. The question is, what do you get by this replica? Maybe you don't understand the simulation, maybe you don't understand biology or things like that. I think it is a worthwhile discussion, really worthwhile discussion. I'm just now reading a philosophical book about the meaning of simulation. What does it mean to simulate? Why should I simulate something? What is the correlation between the simulation and the reality? Are they the same thing or different thing? What do I assume in between? So it's a philosopher from America that wrote a book about philosophy of simulation. So I myself need to learn. I'm just saying that it's now in a high time, because of the capability to simulate everything, atmosphere, atoms, CERN, particles, anything, heavy simulations, heavy data, we're in time of questioning why should we simulate, what is the purpose of simulation. But I don't want to deal with it, I just want to highlight that I'm now very interested in understanding what do I do in terms of this context. You want a little intermission to swallow what I said, to drink water for 5 minutes? Okay. I'll drink too. it's a bit easy. You get all the politicals, all the seminars, solid phase data, high energy, that's okay.
I think they also post the list here, right?
If it's not in
I'm
my
surprised that they put up that stuff in the ICMP though.
there's sometimes
Like the latest.
No, they put a weekly physics thing here. Was it?
if it's not in my calendar, it's not shared. It's not
Oh,
They
okay.
do a weekly physics.
So every week, all the physics seminars, I think they have like a table that they post here
Oh,
on the same
I wonder why though.
board.
So there's a lot of
Yeah, there are a lot of physics people.
There's a lot of people in and students, I guess.
yeah.
Theoretical neuroscience
But it's
is
better
on
to,
the side.
you should just download the form.
Yeah.
Just update.
I'm sure we can find a lot of stuff by near department in physical notes in
All the stuff.
no, that particular topic.
I did so much for Harvard.
Yeah.
It is very,
Yeah, I
very
think
late.
I
Very, very late.
think I remember Han talking about it at least like 2 years ago. With Harvard, which is like very prestigious literature.
Harvard University in Boston.
Right.
If you want to, more theoretically,
Yeah.
Hey, so what happened? Did you go to the last science class or
Yeah,
very
yeah, yeah. So we
how many?
finished talking about instances.
Okay.
I can show you my lectures notes.
Okay.
The next time I one of the students is going to present the optimum model. Oh, and we signed up for
which one?
optimum, associated probability. Which is like made by Hein and Daniel
Right.
and
They sign up for topics and
yeah, there's one not registered anywhere.
oh god.
Chaotic network.
Which one?
Which one? The
Chaotic
88 paper?
network.
I don't know exactly which paper. One on learning, another one dynamics.
One AA.
So do you recommend dynamics or learning?
Well, I know the ones that are very well. And it's in the dark. I don't know which learning.
First learning.
Learning.
First it's not good. Okay, I think last year course from the developer, we also it's the writing of the course, if you want to do presentation.
Okay. So I want to continue fast into the mathematical intuition rather than the biological intuition, some mathematical intuition, and of course Lord Kelvin or William Thomson, a physicist, who developed the cable theory, not for dendrites, not for neurons, but for transmission of electricity under the Atlantic and so on. And he said this sentence, which biologists do not like, of course. He said, I am never content, he's Scottish. So this is a translation to English from his very special Scottish way to say that. I'm never content until I have constructed a mathematical model of what I'm studying. If I succeed in making one I understand, otherwise I do not.
This is what you said.
What?
You said it.
You see, I'm Thompson.
No, you said from you.
Okay, no, but so this is something that it's a controversial issue, because what if you speak to people in the street, of course, you say, what do you mean that you understand only if you have mathematical model? I can understand things. I can understand the DNA without mathematical model. I can understand the heart without mathematical model. I know that it's fun. So
It's an issue, but I'm just bringing it because he's the father of cables. I don't want to discuss what his understanding. I'm just saying that he is the father of cables, and cable theory his cable theory was adopted by Roll to describe dendritic cables, axonal cables, yes? So this is his cable theory, so we should remember Lord Thompson. Again, I don't want, because I lost a little bit time in conversation, I really don't have much time for you this time, so I just want to say that the theory, in general, cable theory or any theory could serve different purposes, and so I'm not going to talk about all these levels of using the theory or using the cable theory, but of course, the first thing that we would like to do, and I want to show you one very good example of using cable theory to better interpret data. I already hinted about the isopotential case, that if I had a better model than the isopotential case of Eccles, the erroneous isopotential assumption of Eccles, I would get a better interpretation. Using my model, I would get a better interpretation of the experimental data. So that's certainly a very important function of theory or of cable theory, but also, of course, I want to provide some predictions. I don't want only to replicate experiments, I want also to predict something that was not measured, upon which the theory is based on. So I will give you one example, really one fast example of that in the context of cable theory. Another important thing of theory in general, and cable theory in particular, is to highlight some key biophysical parameters, because you know you have a complex behavior. There are many parameters that might affect this behavior. The question are, what are the key, what are the fundamental among all these possibly interacting parameters or mechanisms, if you want to call them, what are the key ones that determine your behavior, the behavior you look at. So a good theory, like Hodgkin-Huxley one, for example, or a good model will highlight the key, the minimal key parameters that enables you to replicate, so to speak, the phenomena you try to understand. So this is a Hodgkin-Huxley model. Until the Hodgkin-Huxley model, everybody spoke about spikes. There were many parameters, potassium, sodium, this goes up and this goes down. And eventually they show that a very small set of parameters can explain or can replicate the action potential with some interesting predictions. So this is a very good biophysical model, the Hodgkin-Huxley one. And of course, eventually, especially in this ICNC, LSEC ICNC, we want to understand the model should somehow be able, if it's a good model, successful model, connect the level of biophysics into the level of computation, which is a real jump. Completely different level of description. If you take the biophysics of synapses that look like this, and the Hodgkin-Huxley spike look like this, what is the computational function? Then it means that you have to define computation, or function, or behavior, and you want to connect through your model what you learn at the biophysical level to what you are trying to learn at the computational level, which is really different levels. And this next time when we meet, I'll give you some examples of computation using cable theory, but not today. I just want to highlight this distinction. More, but it's at least this. I want also to say something about Ramon y Cajal, which really is a hero in many ways, but he was also saying some very bad things about theoreticians. Very bad. Because he wrote a very nice little book that you should maybe read if you want to read, An Advice for a Young Scientist. This is the book of Ramon y Cajal, a small book, and he has many good advices. And one of his advices is not to become theoretician. But I could not resist showing it to you, so you can read what he said about us, maybe. But this I like a lot. Basically, the theorist is a lazy person, masquerading as a diligent one. He unconsciously obeys the law of minimal effort because it is easier to fashion a theory than to discover a phenomenon. First of all, it's written beautifully, you must admit. Lazy person. Some people took it very hard. I consider myself semi-theoretician, and being lazy, I think, is the best thing. So it's a compliment for me to be lazy. But I don't think he thought that it's a compliment. But he said other things about the division. It's very interesting because this is written at the time, it's 1905. 1905. This is the magical year of theory of Einstein, but he didn't know that. So maybe today you would think differently. I don't know. So he said many things. You may agree with it or not. But anyway, he wrote this beautiful advice for a young scientist. Okay. So cable theory, you know the cable equation, so I'm not going to repeat it. So this is a current equation, which shows you the axial current, the membrane current, which is at any point x is equal to 0, because every current that comes from the left, so to speak, of the cable can either go out of the cable through the membrane or can continually axially from this point onward. So this is the second derivative. So this is the cable equation. This is a normalized cable equation. So this is the cable equation I mentioned before, and I don't want to go into the intuition of this cable equation, but I gave you the intuition, yes? The axial resistance can split either to membrane current, sorry, the axial current at any point can either split to membrane current or continue straight forward, and at this point the sum is 0, sort of Kirchhoff law. So this is this equation. And one thing that Roll started to show, and this I'm sure you know, but again, tell me if you know too much, so I'm going through, that the solution for the cable equation, for example in the steady state case, depends of course on the boundary conditions for this equation. So if, for example, the boundary is an infinite cable, if it's an infinite passive cable, then the voltage, the steady state voltage, the solution for this equation, yes, is exponential with distance. This is this exponential decay when the cable is infinite. But when the cable is not infinite, which is the case with dendrite, and if the condition is of a sealed end boundary condition, which is this condition, so if the derivative dv dx, which is proportional to the axial current at the end, if the end is sealed, so there is no axial current at the end, then the solution is not anymore exponential, but it's hyperbolic, and it looks like this. So for example, if my cable length was 1 sealed at the end, then my voltage attenuation would be shallower than in the exponential case. Okay? Why is it important? And of course, so the boundary, so if I inject current here and there is a certain boundary here, like a branching boundary, and some boundary, it could be sealed, it could be killed, and so closed circuit or open circuit, I can, or a branch, shunted boundary conditions. So the dendritic tree imposes boundary conditions on the current flowing this direction, or if you want, on the current flowing this direction. At this point, there is a boundary condition that depends on the structure. So the structure imposes boundary condition on the current flow, on the synaptic current that goes from the dendrite to the soma. And this is one of the most shocking, so to speak, theoretical result of Roll, really shocking at that time. So Roll computed analytically with Rinzel the case of a very special symmetrical tree, and I'll say a second about the dissymmetry. So the current is a steady state current injected to this point. So this is the distal sibling passive thinnest branch, 4-order branch, or third, 0, 1, 2, 3. The length of each one of them is 0.25 lambda. So overall there is a 1 lambda length from the stoma to the end. Everything is symmetrical, so these two daughter branches are symmetrical, and these two daughter branches are symmetrical, and everything is symmetrical. We can speak about symmetry, and it's a sealed end at the end, so the end of the dendrite are sealed with a cup, yes, it's sealed, so to speak, with a membrane, and membrane is enough small to consider it as a sealed end, and they computed voltage attenuation, the voltage profile, from the point of injection onto the tree. Okay, so here is the input, and locally you get a lot of voltage, if you want to put it this way. So locally, because of the local large input resistance, because of this thin grafted dendrite, you will get large voltage. There is a very steep voltage attenuation here, from here to here, very steep voltage attenuation, and very shallow voltage attenuation to the sibling branch. Although these two branches are completely identical, yes, there is no any difference between these two branches. It's only the input side is different, yes? Because the input is only here, there is no input there. So this was a shocking thing for biologists, not for physicists, for biologists. Why?
The signal that goes back out to the
Sorry?
That means the signal that goes back out to S?
Yes, so the current, you inject current here, the current flows all over, wherever it can it goes, depending on the resistance along the way, and of course it also goes sideways. That's what would have happened if you had a synapse here. It will generate local EPSP, and this EPSP will attenuate here, will attenuate there, and will also attenuate side branch. And in this case, it's a steady state EPSP, so to speak. Yeah, it's a steady state. So the voltage attenuates to this point. The current goes sideways, and this is the result of the voltage that is generated by this sideways current. And you can see that it's almost isopotential. This little piece of dendrite is almost isopotential because there is a very little drop of voltage from here to here. So this branch is almost isopotential, but not this branch. Very not isopotential. And so this is the profile. So it was shocking for biologists because biologists thought, I have a piece of 0.25 lambda long dendrite, so that's it. So it attenuates exponentially according to e to the power of x divided by, I don't know what, lambda, and this is 2.5, and so it's 2.5. But they forgot the boundary condition, which is not new to mathematicians, but new to biologists. So the boundary conditions, of course, going this way, the boundary condition this way is very different than the boundary condition this way, as I mentioned before, because this is field and boundary condition, and this is branching boundary condition. It's like water flowing into a pipe that goes like this, and by the end, in this case, the water flows onto the wall. It's not the same boundary condition, although the length is the same electrically. So this just shows that the equation is solved depending on the boundary condition. But anyway, it means that you have locally, you have asymmetry in the attenuation of voltage in the tree, very strong asymmetry. And the big attenuation is from where the synapse is into the next branch and so on, until you go into the soma. So this is the local generation of voltage. At the soma you would see this. Another surprise, this is a big surprise for Eccles, is the following. Suppose I would take the same electrode, and instead of injecting the current here, I would inject the current directly to the soma. Same current, the same synapse. Instead of being here, I would inject the synapse here, with the steady state synapse. This is what I would get. This is the dotted line. You can see the very small difference, 50%, maybe not so small, but 50% difference between the local voltage at the soma that would be generated if I would inject the current directly to the soma, as compared to the case that I take the same current away from the soma and injecting it there. At the soma, I see 50%, not very big difference in the steady state case. Why is it so, for Eccles it was a very big problem, because Eccles actually, when he wrote, why did he ignore dendrite, he said the following sentence. He said all the synapses near the soma could be considered to be at the soma. All the synapses away from the soma are away from the soma, so they don't affect the soma. This was his kind of argumentation. But you can see that the distal synapses do have an effect on the soma, and a significant effect relative to the somatic EPSP, direct EPSP in the steady state.
You can reduce the dendritic tree. You just have to know the number of synapses on the dendrite.
And where they are located.
No, but if they reach more or less the same effect on the soma, you don't care.
Yeah, you're right. So one way that the physicist should do it, there is more complexity, but you're right. If I knew that I lose, let's say, 50%, let's say, because I'm here at the soma, I lose 50% of the charge, let's call it like this, so I will just put all the synapses here, and in an upscale by some factor, the factor will depend on the location of the synapse. So there is a factor. So yes, for the distal one, the factor will be 2. For this one, the factor will be 1.5. And for the somatic one, it will be 1. I could do that in the passive case. We'll discuss the nonlinear case later. But you're right, yes. So I just want to highlight that my summary sentence for this is that if you look at the charge, which is actually the integral below the EPSP, or proportional to the integral below the EPSP. And the integral behaves exactly like the steady state case. integral below the EPSP. The time integral of the EPSP behaves like the steady state. The time integral, about 50% of distant synapses, the time integral of the cell body will be 50% of what it would have been if you would inject it directly to the cell body. Meaning that most of the charge, or 50% of the charge injected here, will eventually arrive to the cell body. It may take time, but if you take the integral, 50% of the charge injected by this excitatory synapse will be seen at the soma. That's what I'm trying to summarize. So that's the intuition I wanted to bring here. Why? Because for the charge, if I was a particle, it would be actually easier for me to reach here. There is less resistance this direction, just because of dimension. It's more difficult for me to go there. It's more difficult for me to go there just because of dimension. So I prefer, so to speak, to flow like this. So I lose something along the way out of the membrane to other side branches, and I lose them to ground. I lose them to ground, but most of the charge, or 50% of the charge, will be arriving. Okay? So that's an important thing to remember. This is in the passive linear case, but at least this was another surprise, yes, from cable theory. But I want to also highlight another surprise because of time limitation, and I'll give you to ask questions. I want also to discuss the transient case, because I spoke about steady state. But you know that synapses take transient. And so you can solve the cable equation for the transient case. So I inject now a transient current, not a steady state current, a transient current, and I look at the behavior of the voltage to solve the cable equation for x and t. So let's say that I inject a delta function at x equals 0, and I look at the EPSP along the cable, away from the injection here, 0.5 lambda away from the injection, 1 lambda away from the injection, and so on. This is an infinite cable in this case. And you can see, of course, that there is a velocity for the propagation. It takes time for the signal, because of the RC circuit along the cable, it takes time for the peak to reach as you go away. So there is a velocity of the peak, and of course there is attenuation of the peak or of the integral. And you can prove mathematically that if you go to the infinite distance from, so asymptotically the velocity of the peak in passive cable is 2 lambda over tau. So near the injection point the velocity is faster, but as you go away the velocity is asymptotically converging into this constant velocity of the peak away from the injection point. So that's the velocity of EPSP in an infinite cable, not in dendrite. This is 2 lambda over tau. So if you want to take example, let's say lambda is 1 millimeter, let's say, and let's say the tau is, I don't know what, 20 milliseconds, you can compute the velocity of the EPSP. You can see by this computation, which I'm not going to return because I want to give you insights and not too much mathematics, I want to tell you that if you compute how long does it take for a synaptic input that starts, let's say, at a distant part of the pyramidal circuit to reach, how long does it take for the peak of the EPSP that was generated here to reach the soma, you can do this approximation. You can also compute it more directly, and you can see that this is a big delay in terms of neurons. Yes? So for example, it could be 10 milliseconds delay from the distant synapse to the soma. So the input is there now. It takes 10 milliseconds for the EPSP peak to appear at the soma. It's a huge delay in terms of neurons, yes? It's a big delay. Axonal propagation is much faster. The synaptic delay, the jump, so to speak, between one side to the other is 1 millisecond. So 10 milliseconds is a lot. So I can summarize by saying that dendrites impose a significant delay between the input arriving to the dendrite and the EPSP appearing at the soma, depending where the synapse is, of course. If it's a closed synapse to the soma, then the delay is smaller. But most synapses are not near the soma, you understand that. So dendrites are significant delay line when I say significant, is in the terms of membrane time constant, of tau. Significant delay. The most significant delay of a neuron as a unit is the dendritic delay. So there is dendritic delay, there is axonal delay, there is synaptic delay. Dendrites are the most significant delay line for distant synapses. So this came from cable theory. Another thing that came from cable theory, and this is really, I mean, this is the highlight of what I want to show you as a good model. This is the following. So this is a paper by Roll from '67. And you're probably not aware of it, the people recorded from spinal cord, Eccles was recorded from spinal cord for many, many years and got Nobel Prize for his really beautiful recording. He was the first to impale, one of the first to impale with electrode intracellular into this big cell, the alpha motor neuron in the spinal cord. And when he recorded the cell body of the motor neuron and activated specific axons, recording the EPSP in the cell body, he saw different shapes of EPSP. Some of the EPSPs in the cell body were fast. When he activated other axons, called alpha motor neuron axon, 1A input, it is called 1A input. When he activated this axon, he sometimes see this EPSP. When he activated this axon, he sometimes found this EPSP. So there was a range of shapes. I'm talking about shapes. In this case, normalized shapes of EPSP at the cell body of the motor neuron. Now Eccles, now this is the extreme case of a wrong model that can give you a completely wrong result. He said, because it's an isopotential cell, this is my assumption, because I see different shapes of EPSP, it must mean that each of these inputs must activate different receptors. Time constant of the receptors of the synapse must be different. This is a slow synapse. This is a fast synapse. I mean the kinetics of the synapse. Different receptors. That was his claim. That was the interpretation. Because otherwise, if it was an isopotential cell and you see different EPSPs, what else can it be? It must be that the input, sometimes the input is slow input, sometimes it's a fast input. Maybe NMDA input, we didn't speak about this NMDA. NMDA input versus AMPA input. And this is again what Roel said. He said, just a second, I have a structure, non-isopotential structure. Some synapses are here, some synapses are here, some synapses are here, excitatory synapses. Could the structure give me difference in shapes? You already know yes. I just showed you before that the propagation of the signal along the cable changes the shape. So he said all this, it's the same synapses, exactly the same synapses. They are just by chance. Some of them are located here, some of them are located here. And so the faster one is one that is located near the soma, and the slower one is the one that comes from distance. That was Roll's interpretation. It was a big fight. This means that either there are different type of synapses, or it's the location that determines the shape. It's a big controversy. Of course, in this case, Roll won. There is a whole history to this controversy, and eventually we know that all this 1A input to the dendrites of spinal cord alpha motor neurons are the same synapses. They are just distributed all over. Not only that, but using Roll cable theory, just looking at the shape, you can see where the synapse come from. Exactly. You say, if this is the shape, and actually he called it the shape index, so he computed that if you have a particular input and the rise time or the time to peak is this, and the width, the half width is this, then this synapse should come from 0.2 lambda or 0.5 lambda away from the soma. And if the rise time is this and the half width is this, it should come from 0.8 lambda from the soma. So he already, by sitting at the soma, just looking at the shape of the EPSP, assuming that it's a cable with a particular property, I can already envision, or I can predict, where the synapse comes from. Now, when I say when the synapse comes from, Of course, I don't know if it comes from this dendrite or this dendrite or this dendrite. I know only that